The topic we're discussing today is cloning.
Cloning has been practiced since 1970. There are 3 ways to cloning. DNA and gene cloning, which is used to create a large number of copies of a gene or other pieces of DNA. So basically, DNA cloning is used to figure out what may affect the DNA and what causes mutations in DNA. Secondly, we have reproductive cloning, which is the process of making a genetically identical copy of an organism. Lastly, we have therapeutic cloning. In many other words, cloning for medical purposes, which has the potential to benefit large numbers of people. An example would be stem cells cloned from a person with a disease that could be grown in culture and studied to help researchers understand the disease and develop treatments.
The pros of cloning is maintaining good DNA in animal reproduction, cloning genetically modified animals, for example, xenotransplantation, which is to avoid tissue rejection, and insulin producers. Another pro for cloning is producing whole organs and healthy cells for transplantation, to reduce the number of organ donors, test drugs to understand diseases, to find cures to diseases, and also to save certain endangered species from extinction.
Some of the cons of cloning are that cloning can lead to failures in the course of pregnancy or some sort of birth defects. For example, a cow can be born with 2 heads. A tremendous number of animals are used to produce each clone. Because 99% of cloning attempts fail to produce a healthy clone animal, thousands of embryos and hundreds of egg donors and surrogate mothers are used in cloning ventures. Cloning can reduce genetic diversity since cloning just uses 1 copy of individual's DNA. Cloning involves invasive and painful procedures to animals. Furthermore, cloning is expensive and far from certain. The cost of cloning makes its use in farming and basic research economically infeasible. Also, meats of cloned animals are not yet tested for safety. The meat of cloned animals are not yet tested as a safe food source, so consuming those products may lead to harmful results in the future. Lastly, cloning can cause faster aging or even premature death to animals because when harvesting cells from a donor, raise them to embryonic stage in the petri dish and then place them in the surrogate to grow to term, there is a possibility that the age of the donor could be imprinted on the growing embryo.
Currently, scientists are moving closer to their goal of creating stem cells perfectly matched to patients' DNA in order to treat diseases. Researchers are even looking into resurrecting the dead from DNA found in graveyards. Scientists are also working more on cloning animals. For example, the FDA has approved researchers to make clones of animals with agricultural traits such as the production of milk and lean meat. The cloning of animals is fueling researchers to think that the cloning in humans are the next big thing.
Thank you. I hope you learned a lot about cloning.